Hi, welcome everyone. We'll get started in just a minute. Hey, welcome everyone. We're so glad you could join us for today's webinar with this year's winners of the 2025 Ocean Innovator Awards. My name is Sarah Carr, and I would like to welcome on behalf of OCTO, Open Communications for the Ocean, which is co-hosting today's webinar with Sea and Shore Solutions. Um, OCTO is an NGO that connects ocean professionals to the knowledge and networks they need through a webinar series and our MPA Help, EBM Help, and Ocean Plastic List communities. OCTO also provides custom consulting services. We'll be posting a recording of today's webinar at www.octogroup.org/webinars later today. Um, so thank you all for being here, and I'll turn the floor over to Sea and Shore Solutions, their team right now, for further welcome and instructions.
Sarah, and hello everyone. Thank you so much for joining us at this year's Ocean Innovator Award webinar featuring our amazing 2025 winners. My name is Deanna Carciolo, and I'm the Chief Operating Officer and co-founder of Sea and Shore Solutions, or SNS. SNS is honored to be hosting this award and co-hosting the award webinar with the OCTO group here today. So to quickly kick us off with some quick housekeeping, all attendees are muted with videos turned off throughout the webinar, and closed captioning is available by clicking on the CC button at the bottom of your screen. There will be an opportunity for Q&A with the winners at the end of presentations, so please send any questions through the chat function at any time throughout the webinar, and we'll be collecting those for that Q&A panel. And finally, if you do run into any technical issues, you've already heard from Sarah Carr, you can message her directly in the chat or email her directly at sarah@octogroup.org. So SNS is a women-owned environmental consulting firm focused on creating and implementing innovative solutions for coastal, marine, and estuarine spaces. Our team provides quality, innovative solutions that bridge the gap between best available science and application. As a company, our work is informed by 5 core values: collaboration, strategy, science, innovation, and outcomes. And each year we run the Ocean Innovator Award as a way to dig into our innovation value. This award serves to increase the visibility of high quality, innovative solutions and to support early career professionals. SNS hosts this competition yearly, seeking creative application from these early professionals in coastal, ocean, and estuarine fields that showcase innovative science or projects that contribute to salient ocean topics. Projects can be at any stage of the process to apply, and applications are reviewed by a panel of experts with diverse specialties within the field. And finally, winners are awarded up to $2,000 and are showcased in this webinar. So there is our next competition will be opening in February 2026, so there's time to get involved. And I invite everybody to use the link in the chat to add yourself to the OIA email list so you're notified when the next award cycle is announced. And please feel free to share this link with anybody else who might be interested. And with that, here are our amazing 2025 Ocean Innovators: Jessi Florendo, Elizabeth Farquhar, and Patrick Nichols. It's my honor to introduce our first speaker, Jessi, as they get their slides up. So Jessi is a program coordinator and marine restoration practitioner at the Puget Sound Restoration Fund in Washington State. Committed to the restoration of both Olympia oysters and bull kelp in the coastal Salish Sea waters, they're part of one small but mighty team that develops, implements, monitors, and shares findings on diverse restoration aquaculture approaches for renewing these habitat engineers. As a coordinator, they manage a small portfolio of novel kelp restoration projects and work closely with the Kelp Lab team on innovating and testing kelp growth methods. When duty calls, you can find Jessi anywhere from the office in Seattle to a muddy tidal flat or a paddleboard on the Puget Sound. And finally, Jessi has a Bachelor's of Science in Marine Biology from the University of Rhode Island and a Master's of Marine Affairs from the School of Marine and Environmental Affairs at the University of Washington in Seattle. It's my pleasure to hand it right on over to you, Jessi. Take it away.
Much. Good morning and good afternoon, everyone here. My name is Jessi Florendo, like Deanna's very kind introduction, and I am the program coordinator at Puget Sound Restoration Fund in Washington State. I am so honored to get to share with you all today one of our projects for kelp restoration, which we call kelp gardening. While PSRF is involved in the restoration of different marine species across Coast Salish waters, the primary species that is involved in our kelp restoration work is bull kelp. Scientific name Nereocystis leuciana, this brown macroalgae species ranges from as far south as Baja to as far north as Alaska. Bull kelp is considered a canopy kelp, meaning that it rises all the way to the surface from the seafloor. When dense groups of individuals grow together, bull kelp can form a remarkable habitat called a kelp forest, which serves so many different purposes. As the home to abundant fish and invertebrate species, kelp forests protect juveniles from predators, provide pH refuge from ocean acidification, buffer strong hydrodynamic forces like currents and storms, and even act as food for some grazer species. Bull kelp is important to people, both as a food source and a building material to indigenous people since time immemorial, as well as providing recreational opportunities for watergoers and divers. Finally, bull kelp plays a significant role in cycling nutrients, from producing oxygen as a byproduct of photosynthesis, mitigating nitrogen blooms, which contribute to eutrophication, and even taking in and storing carbon as part of its biomass. In light of this important role of bull kelp to create habitat and contribute to the well-being of marine ecosystems, this species is in decline in different regions throughout its total distribution. In Washington, state agencies have documented a significant loss of bull kelp forests within Puget Sound, with 63% loss in the southernmost extent of the sound when comparing current linear extents to maps from 1878, which increases to over 90% loss in some distinct areas. The cause of this decline is likely due to multiple factors. Abiotic factors contributing to bull kelp decline include different changes in water quality, like warming ocean temperatures or increased turbidity from heightened storm forces. Changes from biotic factors like competition from invasive seaweeds or overgrazing by different grazer species like crabs likely also contribute. Both biotic and abiotic factors are also created and exacerbated by changes of anthropogenic origin, such as shoreline development, which changes sedimentation or increases nutrient runoff. PSRF has been involved in kelp restoration from the early 2010s, which includes stewarding our enhancement site where we've grown bull kelp since 2019. These years of testing methods and learning best practices has revealed the following key hurdles to major kelp restoration projects. One, that permitting from regulators is an extremely cumbersome process, including nearly years of discussions across permitting agencies and the financial burden to coordinate the acquisition of all the proper permits. Two, that kelp restoration sites often require intensive infrastructure to support the growth of dense kelp forests that are able to self-propagate year after year. And three, that the work requires specific staffing, including scientific divers, a boat and captain to be able to access our sites, and training on specific protocols to properly evaluate restoration objectives. While all of this is burdensome to the act of designing a kelp restoration project, it also means that the general community is often left out of the equation, drawn in when we are able to present findings or offered photos and videos when the opportunity presents itself. To overcome these hurdles and draw community members into the work itself, myself and a team of PSRF scientists have piloted a novel project we call kelp gardening. Low cost of entry and permissible efforts to grow bull kelp that is in active collaboration with our community of kelp champions. To begin to design such a project, our team met with regulators and tribes throughout Washington, including the Department of Fish and Wildlife, Department of Natural Resources, and the Suquamish tribe in this first iteration of our work. Through open communication with regulatory agencies, we learned how a kelp restoration project could proceed without flagging the need for a permit or other similar regulatory action. Permitters would allow the use of materials that would break down naturally in seawater, the deployment of seeded line onto existing structures, such as a natural boulder or like an abandoned structure, and the use of a genetic source from a nearby kelp bed to avoid impacting wild kelp. Additionally, we were not allowed to grow kelp for harvest, agreeing that the sole purpose of this pilot is for marine restoration purposes. With the regulatory expectations set, my team set our sights on selecting sites, identifying the locations where we could undertake kelp gardening. We chose to begin this pilot local to Bainbridge Island, where one of our offices is located, as PSRF is really interwoven into the community there. Site selection was informed by meeting with Bainbridge community members who directly weighed in on where they knew kelp used to grow, tapping into that local knowledge, as well as pairing it with the use of a habitat suitability tool, a GIS map you see here, developed by a previous PSRF staff member. Through these conversations with community, we initially chose 4 sites in 2024, which was up to 5 this past year. Next, most important to selecting the site was developing methods that built on the best practices we've developed from years of kelp restoration aquaculture that still fit within the restrictions set by regulators. I led a lot of the technical research and development that you see on the screen here. For investigations into biodegradable twine, while we had originally trialed a number of different plant-based twines, in 2025 and moving forward, we've opted to go with cotton and wool, which have distinct research backing their viability as a substrate for kelp seed. Second, due to the seed bank, which PSRF houses and maintains, which has genetic sources of kelp from across Puget Sound, we are able to grow kelp from nearby Vashon Island, which is close to Bainbridge Island. Third, we tested novel means of growing kelp seed on our cotton and wool twine, leading different experiments in our off-season to trial the different methods for growing kelp onto the twine and optimizing how our lab was set up to produce the best kelpie product available. While community informed the design of this project, it was always in the plan for community to collaborate and co-lead this restoration work. As we planned for outplanting, which is our word for the transfer of lab-grown kelp to our experimental sites, and monitoring, the means by which we watch and determine the success of those sites, community was first and foremost every step of the way. 2 years of piloting kelp gardening on Bainbridge Island has returned different results, all insightful to how we accomplish such a novel approach. Across both years, we were able to steward 3 of our sites directly with community involvement, drawing all folks into the transfer of kelp to substrate and deployment in the field, which is frequently off of paddleboards and kayaks. Due to our immense growth in lab methods, we were actually able to provide seed for 4 sites in 2024 and up to 5 this past year in 2025. Despite this, we have so far observed minimal growth and survivorship across all of our sites after the first year. While that's a hard pill to swallow, not a good result we want to see, this has taught us a lot about what materials are really able to sustainably support kelp, what materials won't break down before the kelp is able to grow onto the natural substrate, as well as informing new important trials to confirm successful kelp production in the lab. Additionally, the planning and implementation of kelp gardening in year 1 taught my team how to simplify our approach in year 2, which led to a more seamless outplant and community events this past February. This is the time of year where bull kelp is an important period of growth, so we won't know how year 2 is faring until later this summer. I'm so lucky to get to share this very important work with you all, and none of it would be possible without the intrepid team of thinkers and tinkerers pictured here and across all of PSRF. Thank you, and I'm really looking forward to hearing any and all of your questions later.
Fantastic. Thank you so much, Jessi. And big reminder to everybody, please add your questions to the chat so that we can ask Jessi all of these amazing follow-ups during the Q&A portion. So now it's my pleasure to introduce our next fantastic winner as they get their slides up. And Elizabeth Farquhar currently works as a research specialist for the Coastal Hazards Lab in the Civil Construction and Environmental Engineering Department at North Carolina State University. In this position, she maintains deployed sensor networks, plans and executes frequent field campaigns, and helps to develop new sensors. She previously received her master's in marine science from University of North Carolina at Wilmington, where she developed a low-cost PCO2 device and honed her technology skills. She's always been fascinated by the marine environment and is most interested in how we can better utilize technology to monitor the ocean for its preservation. And let's see, Elizabeth, I can see your screen, but it looks like it's in presenter mode.
Oh, like you're seeing.
Oh, there we go. That is perfect.
It's good now.
We are all set. Please take it away.
Perfect. Hi, everyone. I'm Liz Farquhar, and as she mentioned, I will be talking about the work that I did during my master's at University of North Carolina at Wilmington, and that work was developing a low-cost device for estimating delta PCO2 in coastal environments. So first, there we go. First, I'm going to show the video that I submitted for this, and you may have to turn up your volume. It's kind of low.
Have you ever wanted to study CO2 concentrations in the coastal environment, but sensors are just too expensive? Perhaps you already have an aquatic PCO2 sensor, but you can only afford to buy one. Now, what's that doing to your sampling spatial resolution? If this sounds like you, then allow me to introduce you to the sea cow. Whether you're trying to preserve blue carbon environments, maybe you're into modeling carbon budgets, or maybe you're even one of those new groovy MCDR groups and need to measure your progress. Whatever it is that you're doing, the sea cow is there for you.
Well, that sounds great and all, but what actually is the sea cow?
Well, the sea cow is a low-cost device made entirely from off-the-shelf parts. It cycles between measuring the PCO2 of the atmosphere and the water that it's sitting in, while other commercial aquatic sensors can easily cost $10,000 or more. The sea cow can be made in parts for about $1,400. That way, you can deploy more of them at one time for the same cost and increase your spatial resolution.
Say, what's this here?
Yes, that's the expanded PTFE membrane that allows CO2 molecules to diffuse into the airstream. That is then measured by a CO2 sensor inside the sea cow. This waterproof design allows the sea cow to be deployed in any conditions, while the cellular capabilities allow you to receive real-time results from anywhere. Our scientists rigorously tested the sea cow to bring you the best quality possible.
Wow, I can't wait to deploy these in the local embayment. I'm gonna make 10 and put them in a seagrass meadow. Thanks, sea cow.
Cool. All right, everyone can still hear me. Perfect. So I hope you guys enjoyed that 2000 style commercial. But now I guess I'm just gonna kind of talk about why I went forward with developing the sea cow. So why do we even care about CO2 in the first place? Well, as I'm sure a lot of you guys know, atmospheric CO2 levels are increasing everywhere, and this has led to lower pH in water, and that's stressful on calcifying organisms. We've also been seeing increased flooding and more intense storms. That background photo there is actually North Carolina. We're also seeing increased sea temperature and subsequent sea level rise globally. and also stress on fish and the communities that rely on them. More importantly, we also really care about the CO2 that's dissolved in seawater, and that's because the ocean absorbs an estimated 26% of CO2 emitted by human activities every year. So the ocean is taking up a lot of CO2. So how do we measure specifically the CO2 that's dissolved in water? So currently there's kind of two methods to do it. You can either calculate it using measurements of total alkalinity, dissolved inorganic carbon, and or pH, or a more common way is to diffuse the CO2 that's dissolved in the water into an airspace, and then you measure that airspace with a sensor. And then these are just two examples on the right. So this first example here, that's headspace equilibrium. And what's happening here is the CO2 that's dissolved in the water, it comes into equilibrium with this airspace here, and then that gets pumped into the CO2 sensor where it's measured. Similarly, over here is a semi-permeable membrane method, and that's when you kind of have some sort of tubing that goes through the water, and the CO2 that's in the water diffuses across that semi-permeable membrane and enters the airstream and also gets measured by a CO2 sensor. And the semi-permeable membrane is kind of what inspired the sea cow's design, which I'll talk about. So I also just wanted to throw this up here. So there's lots of different CO2 systems for measuring various types of things. Some of the kind of high cost, more well-known CO2 systems. So we have the CO2 Pro. That's a delta PCO2 system. That means that it measures CO2 in the water and the air. We also have the LICOR LI50. That one only measures atmospheric CO2. And then we have the SAMI CO2, which only measures aquatic CO2. So you can see that the cost on these ranges quite a bit, and they are pretty expensive. So this bottom section right here is just low-cost CO2 sensors. These are just the sensors themselves. They're not integrated into any system, and that's why they're quite low cost. I just wanted to throw this up here just to show you that if you just get the CO2 sensing mechanism, it's a lot cheaper, but obviously it's not in a system. And why do we even want to use low-cost PCO2 systems? Why not just buy the expensive kind? Well, one of the big kind of motivations for this was to increase our sampling spatial resolution, because if you're able to build a lot more of these for a cheaper cost, you can really increase the area that you're sampling. We also can use CO2 values to calculate CO2 flux. And that just refers to how carbon is moving in and out of systems. And a lot of people are quite interested in that value. And then I'm also especially interested in studying and monitoring important habitats like seagrass. Seagrass is a blue carbon habitat, so it sucks up a lot of carbon, and that's really important to quantify. There's also an increasing number of MCDR groups kind of popping up, and that just stands for marine carbon dioxide removal. These groups are thinking about how do we use the ocean to suck up more carbon so that we can get it out of the atmosphere. And a lot of these groups are a little bit experimental, and they're needing more monitoring to be able to confirm like what they're doing or not. So all of these things have led to the development of the sea cow, which I mentioned in the video, but that is the system for exchange of atmospheric CO2 with water. And the idea behind this machine is that it's kind of a cylinder, and it sits half in water, half out in the air. And then it measures the CO2 in the water and the CO2 of the air. And then you can later take those values and take the difference between them. The advantage of doing a delta PCO2 instead of just water side PCO2 is that with low cost sensors, a lot of times there's kind of sensor drifts. So even if it's reading the same value over time, it'll look like it's not because just drift occurs. But if you take the delta between those, that delta stays the same. So it's able to become a little bit more robust through longer deployments, even if there is some drift, it gets subtracted out. So this is just a, it's kind of like an airflow diagram of the sea cow. Basically, this top side is where the CO2 enters from the air, and it goes through this planar expanded PTFE, which is basically like plumber's tape. And it goes through there and enters this airstream, and then it goes through a series of that's desiccant, and it gets pumped into a K30 sensor where it's actually measured the amount of CO2. These are solenoid valves, so then it switches and it does the same thing, but on the water side. So the CO2 diffuses into this expanded PTFE tubing, and then it goes into this airstream and gets measured also. I'm not going to go through all of this. This is just the circuitry diagram for the C-Cow. Mostly, I just wanted to pop this up here to emphasize that everything that is inside of the C-Cow is an off-the-shelf part that you can buy from lots of like electronic stores like Adafruit or DigiKey or just things like that. So, yep, these are just off-the-shelf parts that you can build. And then one of the major results of this whole experience was just developing the C-Cow itself and trying to make something that works. We did do a lot of characterization experiments throughout the process. I didn't get to like talk about those today, so I'm happy to answer any questions about them. But we compared every single reading from the C-Cows to the Lycor LI850, which was that kind of nicer sensor that I mentioned earlier. And we found that the accuracy is about plus or 2% of what the Lycor reads for the same sample. And this 5 tau here, that just refers to a 99% response time. So that's basically just saying like, what's the response time for the air side and the water side. And then we have the power draw. Dry write is just the desiccant budget and temperature range. I also did want to mention that all of the materials we used, all of the 3D print designs and everything is up on our GitHub page. And we do have a peer-reviewed paper that's about to, it's going through the rounds of editing. So that'll come out eventually. And yeah, that's pretty much it. But I will be taking questions later. And thank you guys for having me.
Fantastic. Thank you so much, Elizabeth. And I'm sure the groovy new MCDR groups love that title as well. That's a fun one.
Yeah,
Quick reminder,
so.
please. Absolutely. Quick reminder to please put questions for Elizabeth into the chat or the Q&A function, and we will get to those after our final presentation, which is courtesy of Patrick Nichols, the first place winner of the 2025 Ocean Innovator Award. Patrick is a PhD candidate in the zoology graduate program at the University of Hawaii at Manoa. His research focuses on developing environmental DNA detection techniques for biosurveillance and coral reef monitoring. Over the past decade, he's worked to advance molecular testing methods that improve how researchers and conservationists track ecological changes and address emerging threats to these fragile ecosystems. His work emphasizes validating eDNA-based approaches while assessing the risks and uncertainties associated with molecular detection tools. Passionate about applying cutting-edge science to the real-world conservation, he collaborates with local nonprofits and stakeholders to integrate molecular data into existing management strategies. I am thrilled to now hand it over to you, Patrick, and it looks like we're seeing the presenter view right now.
Great. Just a second. All right, how about now?
Loading for me. Looks like we're still in presenter view.
Interesting. Sorry.
Patrick, I think if you click display settings up at the top left, then you can swap the slides.
Try one more time. Give me a sec.
All right, coming through loud and clear for us now, Patrick. Take it away.
Seeing the right one? Correct one?
Oh, we were for a second there, but it looks like we're back to presenter view.
All right. Give me a sec. Apologies.
No problem. This is a great time for everyone to please add your questions for Jessi and Elizabeth to the chat or the Q&A function. We're looking forward to getting into more discussion for Patrick's presentation.
Would we want to go ahead and start with one of the questions for the other presenters?
I think I'm ready,
You got it?
actually.
Okay,
I got it.
great.
I
Let's go.
had the screens backwards.
Perfect. There you go. You're all set, Patrick.
Thank you. I hope this is working now. Okay, looks to be good. So thanks again for being here. My name is Patrick Nichols. I am a recent graduate of the zoology PhD program here at the University of Hawaii at Mānoa. And yeah, today I'm going to talk to you about some innovative methods that I've been using in order to track nuisance and invasive species on coral reefs. So this one is called Where in the World is Chondria tumulosa? So just a little bit of background. I have to acknowledge my collaborators and funding sources, though without whom I would not be able to do any of this work. So mahalo to all of you. And this story is basically about the search for a cryptogenic species. So what is a cryptogenic species? Just a little short vocabulary review. So many of the species that you're most likely familiar with are either classified as being native, so naturalized at their current location. Good example of this would be the Pacific purple urchin along the west coast of California, or being invasive, so being introduced and causing some sort of negative impact to us as humans. An example of that is the Indo-Pacific lionfish, which is making its way throughout the Caribbean. But for those that we don't know the origin of, we call those cryptogenic. So for instance, they're found in a location that they are unsure of whether or not they've gotten there via natural pathways or introduced from humans. Example would be oysters in certain regions throughout outside of the eastern seaboard. Not exactly sure, given the historical records, where they have come from. But for organisms that I just don't want to confuse this with terms that I'm going to be using throughout, which is cryptic. That's an organism that is difficult to detect or to find in the environment. So I'll kind of go back and forth between those. But for cryptogenic organisms that we don't know the origin of, they can still be having these negative impacts. So those we call nuisance. So they're behaving invasively, but because we can't pin them as being either native or invasive, we just call them a nuisance. All right, so after that, this story actually unfolds in a place called Papahanaumokuakea Marine National Monument. So this is the northwestern Hawaiian Islands. It's one of the largest and most remote marine reserves in the entire world. It's also home to numerous endemic taxa, some of the most pristine reefs in the region, and it's a World Heritage Site of extreme cultural and ecological significance. So you'd think that it'd be really easy as a scientist here in Hawaii to study this location, and you'd think all the marine scientists are just going and up to these northwestern islands. It's actually really difficult and extremely remote to get up into these locations. And I first want to acknowledge and honor the Kanaka Oiwi who have cared for these islands for generations, and I recognize their enduring relationship with this place, the Hawaiian Kingdom. So the sacred area of Papahanaumokuakea is believed to contain po, which is, according to Hawaiian oral traditions, an entirely divine source of creation, the primordial realm of darkness from which all life has evolved and has come from. And it's essentially the realm of the gods. And that's in contrast to Ao, which is the realm conceived by light and consciousness, and this is associated with humans. So typically no one has inhabited the realm of Po. And even currently, there's only a few managers that are permanently on site in Papahanaumokuakea, and this is just for conservation and management purposes. So if we zoom into the top 3 atolls in Papahanaumokuakea, in 2015, there was an unknown alga that was spotted at Manawai or Pearl and Hermes Atoll. And it was seen covering entire portions of what used to be pristine coral reefs. So you can see here, all this kind of brown stuff on top of the coral reef is that alga. And that meant that it was basically unrecognizable to these managers from previous years of surveying. It formed these extremely dense mats, which they can pull right off of the reef, basically like carpets, and that killed everything underneath. By 2021, it had spread to neighboring Kauai Halani, or Midway, and in 2023 was found at neighboring Holani Ku, or Kure. So this invader was described as a new species of red algae and given the name Chondria tumulosa. And much like the invasive algae that smother a lot of Hawaii's reefs and other tropical locations, it's able to outcompete these native species and again kill everything that's living beneath it. But it wasn't always found growing in such high abundances, especially along its colonization front, where it can be extremely difficult to identify. So even for those of us who are trained in algal identification, you can see that like just a couple little fragments in and among the coral heads, basically blending in as everything else. Not only that, it has the ability to reproduce asexually through a process called vegetative fragmentation. So it breaks apart extremely easily and can spread along ocean currents via rafting, for example, on marine debris, which is plentiful in the reserve. And these fragments have the potential to form entirely new populations elsewhere. So we think that's how it's spreading so quickly. So if we zoom in to Kauai or Midway, ever since the first observations of Chondria tumulosa, researchers have been going up to the monument roughly once per year. That's basically the frequency at which we can access the monument. And we're trying to map out hotspots of abundance, as well as taking habitat conditions such as nutrients, depth data, and the native community that's cohabiting with it. But this is all taking way too long. Again, these expeditions are only happening once per year. By the time we go up the following year, everything is completely different and it's changed. So definitely behind the curve on tracking Chondria tumulosa. So that's really where my research comes in. So I'm using new tools in order to sensitively biomonitor invasive and nuisance species. So I do that using a process called eDNA analysis. So what is eDNA? eDNA is any genetic material that we're collecting from the environment. So all organisms shed DNA, usually in the form of cells or feces and gametes. And by analyzing it, we can get a snapshot of the biodiversity that's in any given area. So much like a forensics team at a crime scene, we can trace the DNA back to whoever was present at our study site. So to assist managers in detecting the nuisance Chondria tumulosa, I developed a set of custom DNA primers that target only Chondria tumulosa. I then spent the next year or so testing these in the lab and on water samples that were brought back to me from the monument, making sure that they're reliable so that they're only detecting the species that we're interested in, nothing else, and that it's extremely sensitive, making sure that we can detect its DNA even when the organism itself is extremely rare. So I'm going to skip the boring details of how we tested all of this in the lab and kind of just go through what we found in the field. So our analysis of eDNA from the 3 atolls where we know Chondria was present pretty much perfectly matched the estimates that we got from visual scuba surveys. So having the divers meticulously go through our sites. So that was a good news because we could just collect these water samples and get the same answer as you would if you were scuba diving. So we also tested eDNA for Chondria from water samples where we don't know if Chondria is showing up yet. So that was throughout the rest of the archipelago at Kapo, Lalo, and the main Hawaiian island. So Oahu, where I'm located, is the busiest and most populated island in the Hawaiian archipelago. And we haven't had any detections further south, which is good. And that makes sense because the prevailing ocean currents actually kind of push everything up to the northwest. So kind of keeping all of these fragments from making their way further south. But managers were extremely worried that this inadvertent transport of fragments could be occurring to the main Hawaiian islands via shipping, these boats that are transiting to and from the reserve, as well as on marine debris, which could wash up down here as well. So luckily this test is a really good way that we can now estimate Chondria presence absence at new sites just by using these collections of water. And what we found is that it's likely not present at the ports and harbors, which berth the expedition vessels that go to and from the monument, which was basically where we expect it to show up first. And importantly, we were able to do this without the need to dive, as I said. So you can imagine in places like Pearl Harbor, it would be extremely prohibitive. I'm sure the Navy would hate us going in there trying to dive. Instead, we can just collect water and determine really quickly whether or not this nuisance species is present. But we still need routine monitoring in order to ensure that it's not spreading throughout the rest of the archipelago. So again, this was just one kind of snapshot. This has to be done over time in order to be an effective early monitoring tool. So I developed a species-specific test. I made sure that it was reliable and sensitive, and in theory, it should be extremely simple for anyone to do, but that was actually not the case at all. So if anyone's ever filtered water before, it's really a pain in the ass. It takes a long time, a lot of equipment, and really easy to contaminate everything. So I'm going to just give you an example of exactly how difficult this is. So to give you, ooh, may have frozen. Can you see my screen? Anyone
I
see
can
the
see
screen?
your screen.
Is it stuck on the same slide?
I believe so, yes.
You could just try backing out of it and starting it again.
All right, I'll do that. All right, can you see my screen again?
Yes, we can.
Great. Moving on, hopefully. All right. So to give you an idea of how difficult it is to collect data in Papahanaumokuakea, so these expeditions are heading to the monument once per year, and this costs roughly $15,000 to $20,000 per day on these ships. And that means that there's a small number of crew that are conducting numerous research tasks all at once. But these methods require specialized equipment such as pumps, power supplies, liquid nitrogen, all this stuff to collect these samples. Not to mention a dedicated laboratory space, which we were fortunate enough to be able to secure to the deck of the ship in these makeshift containers. However, that's really not ideal to do this lab work on a moving, in a container on a moving ship. And furthermore, it meant that these samples had to then come back to the University of Hawaii, where I was able to process them, meaning that our detections were being delayed by weeks to months after the ship had come back to port, which again is just making it harder for these managers to stay ahead of this invasive spread. So I began to think if there was a way that we could make this approach easier for managers in extreme locations, such as Papahanaumokuakea. So to do this, I turned to a relatively new method that's called passive eDNA sampling. So instead of pumping large volumes of water through the filter membranes, we're simply deploying these membranes directly into the water column and allowing the DNA to adhere to it over time. So this can circumvent the collection of bulky water samples, the use of expensive filtration equipment, and the expertise that's required to run all of that. So this is a passive eDNA sampler that I designed in order to be extremely cost effective. It uses materials that can be sourced from just about anywhere. Just to give you an idea, the membrane we're using is a cotton pad, drugstore cotton pad or makeup remover sort of thing. And that costs roughly 3 cents per unit versus $2 per unit for the research grade analytical membranes we were using in the past. But because this method doesn't rely on pumping large volumes of water in order to concentrate DNA, we weren't sure as to whether or not this would be sensitive enough to detect Chondria tumulosa. So last summer, I was fortunate enough to take a quick trip to Kauai Halani, or Midway, and that was my first time to Papahanaumokuakea and first time seeing this organism. And I was able to test out this passive methodology at field sites where we knew Chondria tumulosa was present. So I only had 3 days, and that meant that we had to make sure that this method was working in order to get a bunch of samples in that short period of time. So what I found was that by passively filtering, we actually get lower overall concentrations of target DNA. But while these differences were statistically different, they weren't actually ecologically relevant because both methods were just as sensitive, detecting Chondria tumulosa even at all the sites where it was extremely rare, so in less than 1% abundance. And just to give you an idea, that would be like one of those little fragments in and among the coral reef. So when most people would be extremely unlikely to detect it, this passive methodology still was able to do so. But more importantly, this passive method required fewer steps, less equipment, and that meant that I could collect many more replicate samples in such a short period of time and increase our inferences. So overall, this test is extremely useful. We're deploying these passive membranes in the water for just 15 minutes, whereas again, filtering water takes up to 30 minutes to an hour in order to process one site. It's also extremely versatile, so we're not only attaching these to these simplistic surface marker buoys, we're attaching them to scuba divers in order to extend the depth and the range of our sites. And you can see those being attached right there. And that also allows them to multitask and collect other sources or types of data while passively collecting eDNA. We also paired this with a portable qPCR machine. So this is the machine that actually gives us our molecular detections. And by using this battery operated handheld unit, we can get our detections in less than 2 hours. So now that we had a working eDNA test that was simple enough for pretty much anyone to use, we wanted to see if we could perhaps use it to figure out where in the world Chondria tumulosa may have come from. So again, it's a cryptogenic species. We don't know where it came from, but it's acting invasively. Can we use eDNA to track it? So where would we even begin? So we know that chondria first appeared at Mānoa, Pearl, and Hermes back in 2015, 2016. So we kind of use these prevailing ocean currents in order to backtrack and turn back the clock and see where would chondria most likely have come from given all of these currents at that time. And if we trace all of these potential sources back to the coastlines where they originated from, we see that there's a lot of places around the Pacific where there's a low probability of producing these chondria fragments that would have ended up at Mānoa. But there are a couple places that stood out among the rest, and those are Japan, the Central Pacific, Johnston and Palmyra Atoll, and the Eastern Pacific, Galapagos and Clipperton. So this gives us a good idea of where we should start using the eDNA tool that I've developed. So that's exactly what we did. We actually had eDNA samples that were collected from across the Pacific, and we used these to test for Chondria tumulosa at locations that are the usual stepping stones into the monument. And while the most likely source locations didn't actually produce any hits, we did actually get an unexpected detection at Majuro in the Marshall Islands. So this past November, we headed down there to investigate, and I would like to share the video that I submitted that goes over this process.
In the heart of the Pacific, vibrant corals teem with life, supporting entire ecosystems. But lurking in these underwater havens is a potential threat. Chondria tumulosa. This red alga, first identified in Papahanaumokuakea Marine National Monument, is known for its ability to smother reefs, forming dense mats that outcompete native species. Using cutting-edge molecular techniques, researchers from the University of Hawaiʻi at Mānoa used environmental DNA, or eDNA, to scan the Pacific for signs of this nuisance species. eDNA works by detecting tiny fragments of genetic material left in the water, offering a fast and efficient way to monitor marine ecosystems. When eDNA detected Chondria tumulosa in Majuro Atoll, a location well outside its known range, the team set out to investigate. Using the eDNA results to guide their efforts, the researchers began their search in the waters where the genetic signal was strongest. And on the very first day, they found it. A patch of Chondria tumulosa growing on coral. This discovery highlights the incredible potential of molecular tools like eDNA. By combining cutting-edge science with traditional fieldwork, researchers can pinpoint the presence of nuisance species, map their range, and guide efforts to protect coral reefs. In Majuro, Chondria tumulosa isn't behaving invasively, for now, but its presence raises questions. Where did it come from, and what does its future hold? For these researchers, the work is just beginning. By advancing detection methods and working with local communities, they aim to stay one step ahead of this alga and protect the fragile ecosystems of the Pacific. From Papahanaumokuakea to Majuro, this is science in action, working to understand and preserve the reefs that sustain us all.
All right, so using eDNA, we're pretty much immediately led to the location of Chondria tumulosa, and we found that its growth habit matched, so how we expected it to be growing in and among the coral reef. That matched what we saw in Papahanaumokuakea. Its key morphological characteristics also matched everything that makes Chondria tumulosa unique. And the analysis of its genome has revealed that these populations have been separated for quite some time. So while it's still a little bit of a chicken or the egg scenario, this is still by far the closest known match for this nuisance alga that we have to date. So in conclusion, compared to visually surveying the massive extent of coral reefs, eDNA is extremely sensitive, so we're able to detect trace amounts of DNA. It's also extremely rapid. Again, this publication is still in the review process, and we're able to detect an entirely new population before it has even come out. So yeah, very quick. And it's also cost effective, meaning that we're increasing our field effort and getting many more samples per unit effort. And it's really simple. So we've made these methods cost effective and easy to employ by pretty much anybody, and making sure that it can be done in these extremely remote areas of the Pacific. So with that, thank you all for being here, and special thank you to the organizers who put on this webinar today. Mahalo nui loa. This is my website and contact information if you're curious about some of the other ways that we're using eDNA to monitor coral reefs.
Fantastic. Thank you so much, Patrick. And now we're going to go into a bit of a lightning round of questions. So I invite all of our speakers to come on video, and I'm just going to round robin us really quickly here. Starting with you, Jessi, putting you in the hot seat. So can you share with our attendees a little bit more about how you and the team actually went about planting the kelp?
Yeah, absolutely. I think this is Robert's question that I saw in the chat. And more or less what he describes is the way. We found that the simplest method for getting our kelp out of the lab and into the field has been through the collection of natural boulders that are of a good enough size that they will be able to support the bull kelp without becoming glorified sails when the bull kelp grows and carries the substrate away. So about volleyball-sized boulders, we'll collect those ahead of time, wrap them really well in our substrate, the different biodegradable twines that I described, and then effectively tossing them over the side of kayaks and paddle boards at the locations and the appropriate depth. That is the approach for most of the sites. We are very lucky that one of the sites that is community-led actually has some recreational divers, so they will go out and actually put those more precisely in location, as well as one of our sites is part of PSRF's enhancement site, so our team of divers actually stewards that one through a similar method.
Very comprehensive. It sounds like a blast to put those out. Hopefully not on too many soggy days. So question now for you, Elizabeth. Are there any plans for expanding the deployment of CCOW? And can the CCOW device be purchased by an individual to or obtain the designs for building it out themselves?
Yes. So, wait, sorry. What was the first question? Is there plans to expand? Currently, not really because I finished my master's. They are, however, sitting in my advisor's lab, my old advisor's lab, and there's 4 of them made. So you could probably, I could put my email in the chat. If someone's actually serious, I can like direct you to my old advisor. He has them. And then as for the designs and everything, everything's on GitHub except for the source code. We could work something out though. There was just like the university was trying to get us to do commercialization, but that didn't really pan out. So we weren't like allowed to post the code for a little bit, but I have all the source code and we can work something out, I'm sure. They're not really like for sale right now, but most of their design is out there, and it's been kind of like a weird situation with like patenting, but then also not actually patenting anything. So, yeah, I'm gonna put my email in the chat though, and I like if someone actually has like some more wants to connect on that deeper, I can definitely do that.
Yes, highly recommend. Sensor folks, please reach out to Elizabeth. Great. Now over and back to you, Patrick. How do you see your findings contributing to the future of conservation and management efforts in the region?
Big question there. So yeah, just by showcasing how sensitive these methods can be in order to detect anything that's cryptic or rare, as well as nuisance or invasive, I think that's really the potential that managers can realize by using these tools. If they need to track the spread of things because climate change is moving organisms, you can do it that way. Again, you don't have to be able to dive or even identify anything this way visually. You have no skills in that, that's fine. You just collect the water and we can detect what's in it. So I think that really highlights the conservation potential there.
Fantastic and minimally invasive. It's always a bonus. Fantastic. So back over to you, Jessi, back into the hot seat. Can you share with folks a little bit about how community members interact with your project and how they get involved? Do you find that your team is reaching out more to community members for involvement, or are they coming to you?
Yeah, absolutely. So one of the reasons that we had started this pilot project where we did on Bainbridge Island was because we've already been very involved in the community there. Being local to the area, we know property owners, we know folks who have businesses in the sailing industry and nearby maritime industries. So it was very easy to get started because we already had a bit of a community connection present. That being said, we're continuing to grow this project, and as we think about scaling this project, we are very fortunate and in a very good position that we're connected to folks in the marine space across Puget Sound. So we have networks that we can dig into and pull from. In Washington State, we're very fortunate that some of the water counties have marine resource committees, which have volunteers who are always very excited to get involved. There's local science centers and researchers who have their own community connections. So a lot of what how community has gotten involved so far has been what we have available to us, but we are in a good position to continue to grow and be able to reach out to new folks too.
That's amazing. Thank you. So with that, we are coming up on the end of our webinar here, but we have lots more questions, and we encourage everybody to reach out directly to our awardees to learn more about their amazing and innovative work here. And this is a quick reminder that the Ocean Innovator Award will open back up in February of 2026 for those who might be interested in getting involved. And there's a link in the chat where you can get on that mailing list to get notifications. And with that, I will conclude our Ocean Innovator Award webinar. I thank everybody and our winners for joining and collaborating on this effort this year. Thank you all so much and have a fantastic rest of your weeks.
Thank you.
Thank you.
Thanks, everyone.